Hello and welcome to the Lab of Molecular Neurodegeneration here at Oxford University. We're just going to have a look in the lab at the science that we do. Come on in and follow me. We're now going to have a look at Naroa's research. She actually investigates stem cells that we derive from controls and patients, and she produces neurons and astrocytes, which are glial cells that she's going to talk about, because neurodegenerative disease is not only about neurons, it's also about glial cells. So let's see what science she's doing today.
My project focuses on the role of glial cells, such as astrocytes and microglia in Parkinson's disease. Many people think that we only have neurons in the brain, but we also have these other cell types that are really important for the neuronal survival and the functioning of the brain. Now I'm gonna be feeding some of the astrocytes. These are over 80 days. They have been cultured for over 80 days, and they have been derived from stem cells that were derived from patients. So I need to feed them every 2-3 days, depending on how fast they are growing. Here, as you can see, I'm giving them some cell meat. And here we have a microscope because we can see the cells just with our eyes because they are really small. So we have to come here and on the screen check every day how they are looking, how they are growing. And I'm gonna show you now some really nice astrocytes, and these are over 80 days. Yeah, here like you can see a really nice and big astrocyte with a lot of processes. And as you can see how they are interacting with other cells nearby. And these other cells are neuronal cells. And as you can see, they are smaller than the astrocytes before. And they have many thin processes to interact with the other cells. Cells are kept at 37 degrees in the incubator. Therefore, when we are looking at them in the microscope and they are room temperature, we have to keep them out here for as short a period of time as possible, because they like, as ourselves, they like to be nice and warm inside the incubator.
We're now going to have a look at Marta's research. She is really interested in lysosomal biology, which is the recycling bin of the cell. The cell also needs to recycle its materials and to take away anything that might be detrimental to it. And so Marta's really interested in looking at this, and she's going to show us how she does it in the lab.
So I'm using primary cultures to study neurodegenerative disorders, and I'm looking particularly at lysosomal pathways. And lysosomes are essentially the bins of the cells. And it's very important for neurons to be very efficient at recycling all their components to ensure that they transmit signals properly. And over time, and particularly in neurodegenerative disorders, this mechanism becomes impaired and dysfunctional. So we focus on ways and avenues to bring this pathway back into function, make it more efficient so that neurons can function properly as they should. We study neuronal cells, but also it's important to look at other types of cells that interact with neurons. And in this case, we're looking at astrocytes particularly, but it is also possible to look at other glial cells. And it's important to take in consideration the interactions between these types of cells and how they might influence lysosomal pathways and mechanisms, particularly their contribution to neurodegenerative disorders. So here in green we can see neuronal cells, and in pink we can see astrocytes. And it's particularly important to look at these two types of cells as they will interact in the brain environment, and they will both contribute to neurodegenerative disorders. And when we focus on lysosomal pathways specifically, it's important to consider their interaction. Even other types of glial cells, as they will contribute differently to the brain environment and therefore to neurodegenerative disorders as well. Now, looking at specifically lysosomal pathways, Both neurons and astrocytes will contribute to disease and dysfunction.
After having a look at Marta's and Naroa's science, we're now going to have a look at a new method to detect tau pathology in Alzheimer's disease. So here we're looking at a control brain. We can see the nice neurons in this brain, and that there's very little to no pathology in the form of very tiny puncta that we've shown can detect early tau aggregates. However, if we look at advanced Alzheimer's disease brain, we can see that there's quite a lot of puncta, which are these little tiny dots that represent aggregates. If we look not too hardly, we can already see tau tangles, which are part of the neuropathological events necessary to actually diagnose Alzheimer's disease. However, the challenge was to try and detect this aggregation and this aberrant clumping very early on, so that when disease is just barely started, we can already find it in patients and use this detection technology to find drugs that will tackle the process early on, and so try to modify disease. And this is a patient at Braak stage 1-2, which means that disease is quite early on, and we can see that there's quite a lot of puncta, even if we don't find a lot of tangles as we scroll around, because it's early in the disease, so tangles are not abundant at this point, and this technique is actually really interesting because it can highlight this very early pathology and allow us to develop drugs in this way to tackle it. So here we have some human tissue, human brain samples, and you can probably follow the S-like structure, which is the hippocampus, in all of these brains. And what we have is a control patient, in Braak stage 1-2, which is really early Alzheimer's disease and advanced Alzheimer's disease. And this can take us back to the ARUK Orange campaign, where we can see that there is quite a lot of cell loss when we look at the tissue that is in the hippocampus and the brain cells that are MB0 versus Braak stage 6 or B6. So we can see that shrinkage and that loss that in the end is the size of an orange, the volume of an orange. ARUK funded this very important project in order to detect early tau aggregation. And tau is a protein involved in Alzheimer's disease, particularly in neurofibrillary tangles. And it's a requirement for the diagnosis of Alzheimer's to have tau pathology. And so what we can see between these brains is that the cell loss between B0 and B6 is dramatic. And we were very interested in detecting tau aggregation early on. So not only in patients where it's advanced, but in patients B1-2 or Braak stage 1-2, where disease has just set on, in order to be able to tackle disease early. Thank you for joining us at the Lab of Molecular Neurodegeneration here at Oxford University. I hope you found it interesting, and we'll hope to see you soon.